So without much further ado, because you are here to hear not about me or our sales organization, but our technology. So I'm very happy and honored to be with two of our scientists, Dr. Greg Michelozzi and Dr. Brian Keppler. So Brian is director of our Translational Science department at Metabolon. So Brian and his team, they support our clients in our global business related to pharma, biotech, but also other applied markets. Brian has been with Metabolon since 2011, so he is really part of the success of the company. And Brian received his PhD by the University of North Carolina, and then he complemented his training with a postdoc at the National Institute of Environmental Health Science in the Research Triangle Park, also in North Carolina. And today, Brian will talk about our technology and why we are so unique in the metabolomics space, and how it can benefit multiple therapeutic areas, both in the view of academic research and industrial applications. So, Brian, the floor is yours.
Thank you, Simone. Like Simone said, it's really our pleasure to be here to communicate our technology, provide some case studies, and really try to communicate the power of metabolomics. You know, like Simone said, I've been with Metabolon since 2011, so just celebrating 12 years there this month. And you know, the reason I've been there so long is I really love what I do. I love the science. I love science in general, but I've really had the pleasure of seeing metabolomics grow and seeing Metabolon's role in really pioneering the field of metabolomics, and beyond that, seeing the way that Metabolon can help our clients really accelerate their research. So today I'll give you a little bit of background. What is metabolomics? Who is Metabolon? And then some case studies really showing the power of what we've been able to do with our clients. So first up, metabolomics, right? We're all familiar with the central dogma of DNA being transcribed into RNA and then translated into proteins, right? Most of you probably do some level of omics already, genomics, transcriptomics, proteomics, and all of those are great. They're great tools to use to accelerate your research, and metabolomics in general, Metabolon we're not trying to supplant or replace any of those. We're trying to take your work, your research a step further. And the reason it takes it a step further is because metabolomics is the next step in that central dogma, right? Metabolites are actually closest to the phenotype of a disease or a treatment, or how any sort of challenge can affect the biological system. So whereas the genome, for example, gives you an idea of what could be, right? It depends on the penetrance, right? Does what could be actually happen, right? So metabolites are actually the end result of all different things, environmental and biological interactions. The metabolome also can then be representative of a lifestyle, right? Does somebody exercise? Does somebody smoke? What do they eat? Where do they live? Are there environmental impacts? And also very important, the microbiome, right? Our microbiome within our gut, but also other places within our body. We interact with it in a very symbiotic and commensal relationship, right? And when we're healthy, our gut metabolizes food and is able to send metabolites throughout the body to elicit good things, right? And when there's a disease state, for example, that causes dysbiosis in the gut, then it starts to put things in make things go wrong, right? So our platform, which I'll go into detail, detects metabolites not only endogenous to biological systems and mammals, humans and other mammals, but also metabolites related to the microbiome. So the space that metabolomics is applicable is really across the full continuum of therapeutic areas. We've worked in hundreds of different disease states, and we really apply our technology to achieve a number of different objectives. It can be simply looking at a healthy individual versus a diseased individual. It can be looking at biomarkers for efficacy after treatment, understanding the microbiome, as I mentioned, optimizing drug development, patient stratification, and really the list goes on and on. As it relates to the drug development pipeline, we also work with clients sending us samples from really the continuum of R&D, from basic science, basic research, just discovery, all the way through preclinical studies, looking at rodents, and then clinical studies as well, looking at humans. And again, the objectives are really endless. We have examples where we've worked in all sorts of spaces, again, looking at biomarkers, mechanism of action, safety and efficacy, toxicity. So just to give you a little bit of background, if any of you have done metabolomics, let's say a long time ago, decades ago, compared to today, metabolomics, the technology was pretty poor. And Metabolon existed 2 decades ago, and we were doing really well. But again, compared to today, it was pretty poor. So if you haven't done it in a while, I encourage you to try out metabolomics again, because it's a totally different ballgame now. Let's see. 15 years ago, Metabolon was only detecting about 200 metabolites with error rates 20-30%. Today, we're detecting probably about 1,200 metabolites in a plasma sample with single-digit RSDs, right? And the 2 graphs up here actually show the trends, the growing acceptance and adoption of metabolomics, looking at publications on metabolites and lipids on the left, and then on the right, the incorporation of metabolomics within multi-omics studies over the different years. And as I said, not just looking at basic discovery research, not just hypothesis generating, but all the way through clinical studies, even using metabolites as clinical endpoints. So you can see the growing list of metabolites and lipids in clinical trials throughout the world. So who is Metabolon? All right, what do we do? I don't think I'm going out on a limb here saying that Metabolon truly is the global leader in metabolomics. Like I said, the reason I've been at Metabolon so long is because we do it the best, we have the best coverage, the best capacity, and the highest quality. So our present library for our global untargeted platform, and that's really our bread and butter. That's usually the first step that researchers take to cast the wide net, look in an untargeted, unbiased fashion to look at anything and everything. So that current library is about 5,400 metabolites, a really unrivaled list in terms of size. As I said, we've worked for about 20 years, and this library of 5,400 metabolites, it leaves no stone unturned, right? We're looking at all metabolic pathways, all super pathways, all sub-pathways. This technology has really allowed us to analyze samples from any species, any tissue type, and throughout the 10,000 plus projects we've done throughout our lifespan at Metabolon, we have about 3,000 publications with our clients, again, in various disease states, various therapeutic areas. So a little bit about the quality of what we produce, right? I said an unrivaled level of capacity and coverage throughout the various pathways, but quality is really the most important, right? So this is a tier-based system shown here. It was actually developed by the Metabolomics Society Initiative, showing someone what is the quality, what is the level of annotation or identification of a given metabolite. And you can be from tier 1 to tier 5, right? Do you only know certain molecular formula? Do you have a tentative structure? Do you know the actual structure? Do you have an actual validated, purified standard that you're comparing it against? And Metabolon, for the most part, lives at tier 1. I'd say 90 to 95% of our metabolites on our global platform are all tier 1, which means we know exactly where they are. We've run all 5,400 metabolites standards across our platform, and we know all the ion features that they produce: retention time, mass, fragmentation, etc. So when we see those same fragmentation patterns, those same ion features in any of your samples, we are 100% confident that it is this specific metabolite. The other 5 to 10% of metabolites on our platform are not tier 1, but tier 2. The only reason they're tier 2 is we don't precisely have a purified standard for those, and when we do report those in your study, we are fairly confident in their identification, but we do give an indication that they are tier 2 presently due to the lack of an internal standard, of a purified standard. And on the right here, you can see a high-level look at those different 70-plus pathways, right? Again, looking at everything, no stone unturned. We are looking at carbohydrate metabolism from energetics, amino acids, and when I say amino acids, it's not just 20 amino acids. Our amino acid superpathway contains about 400 metabolites, right? So it's not just important to say, okay, is my amino acid going up or down, but what is it destined to become? Is it being utilized for protein, for biomass, or is it being broken down for energy, or is it being broken down for some other reason that's not beneficial? Lipid metabolism, nucleotide metabolism, exogenous metabolites, basically any commercially available metabolite that is possible to purchase is in our library, in addition to some that we've made in-house at Metabolon. And along with that quality of identifications, we also have a number of QA steps that we institute at Metabolon. So we are an ISO 9001 accredited lab. We're also CLIA certified and CAP accredited. We also abide by FDA and EMA guidelines, and we also have capabilities for GCP and GCLP for our targeted assays. As it relates to many of you, we are also rigorous data governance program, and we have adherence to GDPR as well. And also a very robust quality control system. Anytime we run one of your studies, we're utilizing internal standards, recovery standards. We're running technical replicates from all of your biological replicates so we can really monitor process and platform variability. So I've talked to you mostly about, on the left-hand side, the global discovery panel, and most of the case studies that I'm going to talk about are going to be data from that global discovery panel. But we also have extensive capabilities in a targeted sense, right? So the global panel is discovery-oriented. We're not running standards for each one of those 5,400 metabolites, so it is semi-quantitative, relative quantitation. And at that level of discovery, that's really all you need, right? So we're providing you with fold changes to give you hypotheses on what to look at maybe in a more targeted approach later on. All of our targeted approaches are fully quantitative. We have some off-the-shelf targeted panels, 20 to 30, I believe now. We've actually launched some new ones this year, and we have plans to launch additional ones next year. Just some examples are up here. A complex lipid targeted panel actually provides an additional 1,100 lipid species. These are fully compositionally and speciation. We also have a bile acid panel, short chain fatty acid panel, insulin resistance targeted panel. Again, I don't have the full list up here, but many off-the-shelf targeted panels. In addition to the ability to develop any custom targeted panel, which is a big part of our business now. So using the discovery panel, if it sends you down a route to say, okay, these are my 5 biomarkers that I want to look at in this species, in this matrix, we can, if we don't have experience in doing those specific metabolites, we can build it from the ground up. And again, we can do it in under whichever regulatory standards you need, simply research use only or GCLP. So this is a quick high-level overview of our process from talking to a client for the first time all the way through delivering data. So my team within the Discovery and Translational Sciences group really likes to be involved on the very front end, the inception of a study design idea, right? We're involved in client conversations with our business development to really understand what your objectives are, right? And the objective is what is going to dictate your study design. So we have a lot of experience in what worked and what hasn't worked. So we like to really share that knowledge so that you can get the most out of your study. We can provide you with sample collection, sample shipment, delivery protocols. And then for the global approach, when samples are received in-house, regardless of really what species or matrix type it is, it's a fairly simple extraction where we're crashing out all RNA and DNA and proteins and really just focusing on those metabolites, typically between 50 to 1,500 Daltons. And each sample is run across, as I've been saying, our global untargeted panel. And that panel is really 4 different methods, and they're all LC-MS methods. And the 4 different methods really are differentiated by the LC columns. So many metabolites are optimized for positive ionization, some for negative ionization, some are polar, right? So using a single method will not allow this panel to be truly global. So we have 4 different methods to really capture the largest breadth of different classes of biochemicals. So one sample run across any one of these assays in the platform is going to generate hundreds of thousands of ion features. And that's where the hard part starts, really. It is deciphering all of these peaks, right? How do I then figure out what metabolite all these peaks correlate to? And that's really a lot of the effort that Metabolon has put in over the last 20 years, and really what differentiates us to be able to do it with high quality, but also with high throughput. So really trying to reduce the data, eliminate the noise, and saying, okay, all these peaks are related to glucose, et cetera, et cetera. It's really transforming the data from an ion-centric approach to a chemocentric approach. And this is really a good point I want to make about differentiating metabolomics, the field of metabolomics. Some people work on an ion-centric manner and some on a chemocentric manner, right? If you're working on an ion-centric manner, you don't precisely know what it is. You might know that it's a 6-carbon sugar, but you don't know which one, right? When you're working on a chemocentric approach, you know exactly which 6-carbon sugar it is. You know it is glucose, right? That speaks again to that tier system that I was talking about earlier, about tier 1 knowing exactly what a metabolite is. So regardless of what level of metabolomics you do, whoever you work with, I implore you to make sure you're getting good quality data. The final step is then delivery of the results. We'll deliver the data to you. We'll perform all the statistics based on your metadata, and then we actually take it a step further. We try to go beyond just delivering data to our clients. We want to deliver knowledge and information, right? Metabolomics is a pretty complex omics to interpret, and people on my team and the people in Metabolon have been doing this for 20 years. So we want to share that experience and help really act as an extension of your team to interpret the data for you, tell you if what we're seeing is typical or if it's atypical, if it's very unique to your study. We want to accelerate your research as fast and as efficiently as possible. So last few slides here before I get into the case studies are going to focus on the microbiome. So that library of 5,400 plus metabolites, as I said, you're not going to see 5,400 metabolites in any given matrix, right? A plasma matrix is one of the more rich matrices. You're going to see about 1,200. The reason you're not going to see 5,400 in any given matrix is because not all 5,400 are mammalian, for example. The majority of them are mammalian, but we also have a lot of plant-related metabolites. We do a lot of ag bio work, but also humans consume plants, right? And we digest them, and the microbiome within our system digests them. So a lot of these metabolites in our library are also microbially derived. And we know they're microbially derived because humans and mammals in general simply don't contain the gene to lead to the protein that can produce them, right? So when we see them in our feces or even recirculated within our circulating biofluid, we know it is microbially derived, and that's how our platform can give insight into changes in microbiome with, let's say, disease or diet change or treatment. And like I said before when I was talking about the central dogma, metabolomics is not meant to supplant metagenomics either, right? Metagenomics has its place and it's extremely informative, but there are some limitations, right? The presence of a bacterial strain or a bacterial load or a community structure within your microbiome, to know that is extremely important, but the presence of those is not the complete picture, right? Metabolomics takes it a step further. Are those species active, right? Are they producing things that are healthy? Are they producing too much, too little, right? Are they being recirculated? And really metabolites, the application of metabolomics in microbiome research is really, it's obvious. It's a no-brainer, right? It should always be done. And the reason is because bacteria literally consume metabolites and they produce metabolites. The only way that the microbiome within our body can communicate with us as a host is through metabolomics, through metabolites. And here a similar graph to what I showed you before, just focusing on publications related to metabolites and metabolomics within the microbiome. And as you can see in the last 5 to 8 years, really seeing a huge spike. I mean, the microbiome field in general has exploded, as it should be, with new technologies, and metabolomics and Metabolon's technology is really at the forefront of shedding light on all these previously unelucidated questions. Here's just a very quick high-level look at some of the publications. We have about a little over 300 publications specific to the microbiome in various disease states, various objectives. So again, the work is well documented in high-impact journals. So I'm going to move into some case studies next. First couple are going to be just really some seminal papers on the microbiome and in metabolomics. So a couple here, just looking at a couple on a single slide. The first one is looking at the fecal microbiome, the fecal metabolome as really a functional readout of the gut microbiome. A really complex characterization of over 1,000 metabolites from 700 individuals in a population health based study, it was actually the Twins UK cohort, revealed that the fecal metabolome was only modestly influenced by host genetics, but actually more largely influenced by gut microbial composition, right? Again, the idea that bacterial composition is extremely important, but it's only part of the answer. The publication on the right, looking at the blood metabolome and how it's able to predict gut microbiome alpha diversity in humans, actually, so Shannon diversity. So 40 diverse plasma metabolites were actually able to predict Shannon diversity. And the importance of this is actually that beyond that metabolomics could do it, but that other omics couldn't. It was actually shown in this paper that wide clinical tests and a set of 263 proteins could not actually achieve the prediction that metabolomics could. This next publication is looking at the structure, function, and diversity of a healthy microbiome. So on the top here, you're looking at the phyla, and on the bottom, you're looking at metabolic pathways. And this study showed that profiling of 242 individuals showed that the diversity and abundance of the microbes was found to vary widely, even among healthy subjects, and that the metagenomic carriage of metabolic pathways was stable among individuals despite variation in community structure. So basically, if you look at individuals that have different microbiome makeup, different bacterial loads, community structure populations, they can still actually have the same metabolomic profiles. And vice versa, people with the same microbiota profiles can have different metabolomic profiles, right? Again, really emphasizing the point, nailing that point home that stopping at metagenomics or genomics in general is not going to give you the full answer, right? It's a partial answer. So getting into a couple case studies specific to the microbiome, specific to therapeutic areas, here's a couple on the CNS, neurology. Parkinson's on the left, Alzheimer's on the right. So on the left, this was looking at complex alterations in human and microbial sulfur metabolism in Parkinson's disease. And it was found that most of the metabolites, most of the pathways were related to oxidative stress, suggesting that oxidative stress was a likely mechanism at which this was happening. They were also microbially derived secondary bile acids, most of them being taurine conjugated, that were associated with both disease severity and Parkinson's disease incidence. Similarly, in Alzheimer's disease, it was found there's an emerging role of the microbiome. Both of these are very important for the gut-brain axis that's being elucidated. So here, looking at serum, significantly lower concentrations of different bile acids, the primary bile acid cholate and the bacterially produced secondary bile acid deoxycholate were actually indicative of a dysfunction in enzymatic activities of gut bacteria. And these changes were strongly associated with cognitive decline in Alzheimer's, and this was actually replicated in serum and brain tissue from an independent cohort. So this next case study is looking at identification of therapeutic interventions and translatable biomarkers. Again, another microbiome-related one, a way in which metabolites, metabolomics and metabolomics technology has really been able to deliver actionable insights in this area. And this one deals with the metabolite imidazole propionate, which is histidine-derived, but it is produced exclusively by bacteria. So the challenge here was that it's known that intestinal microbiota may contribute to metabolic syndrome, type 2 diabetes specifically. So to identify mechanisms specific to this disorder, metabolomics was performed on different blood samples from subjects with type 2 diabetes. And in the far left there, you can see in the bar graph, the far left one is human. So humans with type 2 diabetes or no type 2 diabetes, and you can see that's a plot for imidazole propionate levels, and you can see they're significantly elevated in type 2 diabetics. The one just to the right of that is looking at mice, so germ-free mice versus conventional mice. And you can see when you knock out the microbiome in a mouse, germ-free, you don't have the increased levels of this imidazole propionate, showing that it is, in fact, a microbially derived metabolite. In the line plot in the middle, histidine was given exogenously. It was within patients, and you can see that when it is given exogenously, increased levels of imidazole propionate were being found and identified. And it was actually found based on our data from Metabolon to the client that a novel pathway, a microbiota-derived imidazole propionate, may modulate the mTORC pathway signaling in type 2 diabetes. So this work really helped to identify a novel interaction between diet, because cysteine is an amino acid that's provided by the diet, interaction between that and the microbiome, and metabolite signaling that can regulate both host health and disease pathology. This next case study is really a good one, and I'll start talking about a previous study that's not really part of this case study to set the stage for this one. So this is done in ASD, Autism Spectrum Disorder, and it was found using a mouse model for ASD, and the mouse model was developed, and these mice showed traits of autistic traits, if you will, behavioral and social traits with an ASD phenotype. And through those rodent studies, they found that there are many microbiome-related metabolites that were elevated, lots of phenolic compounds, the primary one being 4-EPS, 4-ethyl phenylsulfate. The client then took this and tried to determine if this was translatable from mice to humans, and that's really what this case study is showing. So the heat map in the middle here is looking at plasma, P plasma, F feces, seeing if the levels of these, if many different metabolites were altered in ASD patients, so ASD children, or TD children, meaning typically developing children. And you can see the top one in red in the plasma, significantly elevated, again, 4-EPS, 4-ethyl phenylsulfate. And the same data, the top right box plot is for 4-EPS, and you can see some of the other microbially derived box plots are showing similar trends. So this led to this client trying to determine, and one other important note to make, in the rodent studies, it was found that providing exogenous 4-EPS to healthy mice actually induced the traits, the ASD traits, right? So it's not just, it is an actually causative metabolite, something produced within the gut, recirculated into the body, affecting the gut-brain axis and inducing these ASD traits. So it is truly a causative metabolite. So the approach, and I'll touch on this very briefly, but it's highlighted in red here at the bottom, was not really to prevent the production of this metabolite as a therapeutic avenue. It was simply to get rid of this metabolite from the system. So basically, the emerging biotech is using this data to develop a gut-selective sequestering agent, right? The treatment is a gut-selective sequestering agent that simply absorbs 4-EPS and these other phenolic compounds that allows them to be excreted so that they can then not be reabsorbed and affect the host in a negative way. This case study is looking at therapeutic opportunity in relapsed leukemia. So really, this is really an interomics case study where metabolomics was able to synergize some genomics finding. And this is work done in ALL, acute lymphocytic leukemia. And ALL is really, the challenge here is that there's poor cure rates of only about 40%. And interestingly, some of those poor rates are related to gene mutations, specifically in a specific gene, NT5C2. About 20% of all relapsed ALL has that mutation. So the question was, well, how are these genetic mutations, how are these patients resisting treatment, right? How are they, what is the mechanism driving the resistance? And through metabolomics, it was found that it's related to nucleotide metabolism, purine metabolism specifically. So within de novo synthesis, IMP, inosine monophosphate, is broken down into inosine, and you can see the levels of IMP are decreased in the mutant, and the levels of inosine are slightly increased, and that's intracellularly. However, when you look extracellularly, it's flipped, right? You have inosine is highly elevated in extracellularly, as is hypoxanthine. So the take-home message really was that the mutants actually shunt these different building blocks for DNA out to the extracellular system, making them vulnerable to other drugs in the pathway. So the approach the target identification that the metabolomics was able to provide was that perhaps targeting IMPDH, the enzyme inosine monophosphate dehydrogenase, the question was, could that overcome resistance and increase survival? And in fact, it did. The diagram on the top right here is really shows a schematic way in which that happens, right? So the sequencing found a resistance mutation. Metabolomics was really key to deciphering the mechanism of that. And the study was really able to demonstrate and add functional layer to this systems approach. A couple more case studies. This one is looking at understanding metabolic reprogramming and identifying combinatorial approaches. So this is done in B-cell lymphoma, and B-cell lymphoma is really challenged by resistance to therapy, similar to the ALL case study. HDAC therapies, HDAC inhibitors are a common approach, histone deacetylase inhibitors. They really have a lower than expected efficiency in treating B-cell lymphomas. And what was performed in this study was a phase 2 trial showing significant alterations in a number of different metabolites. But the focus really, the concentration of those significant changes was really within lipid metabolism, particularly choline. Can't really see it here, but within the oval here is the biosynthetic pathway for phosphatidylcholine membranes, for example. Choline is converted to phosphocholene, then CDP-choline, and then phosphatidylcholine. And it was found that this shift involved inhibition of choline or the phosphoinositol 3 kinase, PI3 kinase pathways, which resulted in increased sensitivity to penobinostat. So rather than using penobinostat or rituximab by themselves, it was shown through this metabolomic study that using a combination was actually the best approach to not only target the typical pathways of this disease, but these pathways in which the resistance is occurring. And this is the last case study before I'll pause for questions. Another resistance mechanism case study, identification of a metabolomic tumor suppressor for metastasis and drug resistance. So EMT, or the epithelial mesenchymal transition, is a common mechanism by which cancer cells evade drug action. You can see some cell figures on the very left from the publication. Parental epithelial cancer cells go through EMT, become mesenchymal cancer cells, and this is really how they're able to metastasize and drive drug resistance. So there's definitely a need for therapeutic interventions to avoid this or prevent it, and it's really a major challenge in this specific cancer therapy. So a systems approach was employed, metabolomics was employed, and it was found that the integrated data pointed to rewiring during that epithelial mesenchymal transition that actually diverted glucose to the TCA cycle, specifically by reducing the expression of PDK4, pyruvate dehydrogenase kinase 4. So again, our metabolomics insight suggested that this enzyme was perturbed, which allowed the client to then go into their transcriptomics proteomics and confirm that. And manipulation of PDK4 controlled the fate of the epithelial mesenchymal transition, promoted drug resistance, and then was shown to be a predictor of poor prognosis in lung cancer. So again, really implicating that this unique metabolic reprogramming event could be identified and elucidated through metabolomics, providing novel therapeutic opportunities.